Hello guys, welcome back to my channel BioLab, and this is Vamsi. And in today's video we will be discussing the NCRT textbook, full textbook of plant growth and development. So I'm telling you guys, I'm really sorry for not uploading the MCQ video of this chapter today evening, and since I haven't still completed the slides, so that I'll be uploading it tomorrow morning as usual, like today only. Yes guys, I'm extremely sorry for that, and hope you have watched the morning's MCQ series video or the preceded video of this on microbes. We have done MCQ series and also NCRT and also detailed videos. Three are completed for microbes. If you complete today this one, we will be completing plant growth regulation or development, the detailed videos and the NCRT. And in tomorrow's morning session, I will be trying to complete the MCQ series or MCQs on this chapter, that is here 30 under 30 MCQs under 30 minutes. So let's start. So in this chapter, the main things that we'll be learning is growth. What is growth? What is the definition of growth? Differentiation, two types of differentiation, dedifferentiation and redifferentiation. We'll be also learning about development, how growth and development are related. Also we'll be seeing, we'll be talking about PGRs or plant growth regulators, photoperiodism and vernalization. These two I haven't covered in our regular videos or detailed videos, so that is why I'll be covering them in this video itself. Yes, we will be trying to complete this video in 1 hour 30 minutes. Let's try. That's our target time, 1.5 hours. Let's try to complete it. For trying or for completing the lecture in 1.5 hours, I require your concentration, full concentration. Get your NCRT, underline the lines. Now let's starting with this is a very important point that we have to remember, is that it is essential and sufficient to know that the development of a mature plant from a zygote, or you can also put it in other way, you can say plant, sorry guys, 1 minute, what's happening there is a lag in video, 1 minute. Yeah, the thing which we have to understand here is plant starts its growth and development from its zygote stage only. From the stage of zygote only the plant starts its journey. The plant starts its journey showing development. So that's the point we have to remember here. That can appear as a question. Remember, this plant starts its development as the zygote only. Okay, and from there, as the plant is getting matured, it develops roots, leaves, branches, flowers, fruits, seeds, and eventually they die. Death is senescence. Okay, now let's move on to the second page. So underline this point in your first page of plant growth and development chapter. I don't know the exact page number in which it is there. I do not have the NCRT textbook, the print textbook with me. I have downloaded from the other the net source. So that is, see this on your screens, you can see a image. So this is the development and germination of a seedling. It is the development of a seedling. First it was a seed which we planted inside the soil. In a seed we have two portions, or we can say two projections. See this is the seed. We have one radical, one plumule. The plumule develops into shoot, becomes a shoot part, the upper part, this one, and the radical becomes into root part, the underground part, this one. We all know this, and as it is a seed, we water it. From the stage of seed we water it, it develops the roots from radicle, it develops shoots from pumule, and this whole thing is guided or given food with the help of cotyledon. Cotyledon provides food. Based upon the number of cotyledons, we have also classified plants into two types, dicots and monocots. Now after this, root forms, shoot forms, this is called a seedling. Or next, that's what happens here. Again here, this here the leaves develop, after this one. Still the cotyledon remains here in these two stages. Here cotyledon remains still. Now this upper part which is developing and germinating is called as epicotyl, and the lower part which is germinating downwards is called as hypocotyl, and now it is grown into a perfect seedling or plantlet. From here the plant continues its journey to spread its branches, stem getting hard, development of bark on it, etc., etc. So now let's understand the meaning of growth. Growth can be defined as, I have already given you the definition, it can be defined as an irreversible permanent increase in the size of an organ, or 1 minute, let me zoom it on little here. Yes, now it's correct. Growth can be defined as an irreversible permanent increase. Irreversible permanent, growth cannot be reversed, it is permanent. Both mean the same, but in your NCERT it is given like that, remember that definition. Increase in the size of an organ or its parts, or even an individual cell. We know that plants and animals are made up of multi cells or many cells. We are multicellular generally. Even if a single cell in our body at the cellular level grows, then the tissue grows, the remaining cells grows, eventually tissue grows, if tissue grows, the other tissues grow, and the organ grows, organism grows. Here any part, if any one part has a permanent irreversible increase in its size, that can be termed as growth, even at the cellular level. This growth is an metabolic process or energy consumption, and also energy breaking. Catabolism and anabolism both occur at a time, and it occurs at the expenditure of energy or release of energy. For example, expansion of a leaf is growth. See, we have a diagram in our NCERT. 1 minute guys, 1 minute. Yes, even the expansion of leaf is also termed as growth. Now, is plant growth determinate or indeterminate, or it is both? Yeah, let's see that. Plant growth plant growth is unique because plants retain the capacity for unlimited growth throughout their life. Why this is very special? Why plant growth is very special? Because they can grow unlimited throughout their life. This is possible only because of the meristems at the extreme ends or extreme locations of the body parts. These meristems have the capacity of dividing and self-perpetuating, or they possess the capacity to divide and self-perpetuate. Okay, the product, however, soon loses the capacity to divide, and such cells make up the plant body. Now these meristems, they possess the ability to divide. The division occurs until a phase of life in meristems, not in plant. These meristems possess the ability to divide, and some meristems get converted into permanent cells. These meristems are dividing juvenile cells. They get converted into young cells, or these are young cells. They get converted into old cells, or permanent cells. As we from our juvenile phase, juvenile age, from 1 year, or you can say 1 month, we are now 15, 16, 18, 20, 24, 35, like that we grow on. As the age is increasing, our body stamina decreases, and at a particular time we look young. So like that only, these meristems which are present at the extreme ends of the plant body convert into permanent cells, and these permanent cells make up the plant body. Now this form of growth in which new cells are being added to the plant body by the activity of meristems is called as open form of growth. Always a new cell is being added to the plant. Always a new cell is being added. This is one cell, for this another cell is being added, two cells. If these are two cells, another cell is being added, three cells, 4, 5, 6, 7, infinite till they die. And this form of growth is termed as open growth. This form of growth is termed as open growth. Now let's see, what are these meristems? These meristems are two. We have already studied them. One, root apical meristem and shoot apical meristem. Why apical? They are present at the apex of the plant. Lateral meristems are there. They are present at the lateral ends of the plant. Root apical and shoot apical meristems. Now let us see these root apical and let's see what are these root apical meristems and shoot apical meristems. See, there is a diagram on the right side of your textbook. In your textbook it is also present. We assume this diagram as a capsule, or we assume the whole plant as a capsule. In this capsule we divide it into half, and the upper half we think it as a shoot, and the lower half we assume it as a root. Now if the upper part is the shoot, then the extreme end of upper part or the shoot are present your shoot apical meristems. This dark black color area is your shoot apical meristem, Now the middle part, this is all the vascular cambium. And what I said, the lateral meristem is this vascular cambium. Lateral meristem is your vascular cambium. This is shoot, this is root. Root is present. Vascular cambium is also present in root, and here this is your extreme part of your root or capsule, and that is your root apical meristem. Now the thing we have to learn is, there are two types of growth. One, primary growth. Two, secondary growth. Primary growth means the plants grow along the principal axis, or the, we can say the perpendicular axis, or you can also call it as elongation. But as we know, in dicotyledonous plants and gymnosperms, these lateral meristems develop later in their lifetime, and these lateral meristems are your vascular cambium and cork cambium, as I said. These which grow later in their lives are called as vascular and cork cambiums in dicotyledonous plants and gymnosperms. Now what happens is, these cork cambium and vascular cambium increase the girth of the plant. If this is the plant, let's remove all this rubbish things which I have drew now. If this is a plant, they increase the plant like this. See, they increase the plant like this. This is how our vascular cambium and cork cambium increase their length. Girth. Increase their girth, not length. Girth. This girth increase is termed as secondary growth. In plants. Can we measure growth? Yes, we can. Definitely we can measure growth. And growth, how we can measure growth? We can measure growth in many ways. One, it is increase in fresh weight or increase in dry weight. What is dry weight? The weight which is present in the plant by after removing whole water. Okay, that is called as dry weight. Length, area, volume, and cell number. If there is an increase in cell number, if the dividing cells divide and increase their number, that is also termed as growth. If the cells increase their volume, that is also termed as growth. If the cells increase their length, that is also termed as growth. If the cells increase their area, that is also termed as growth. If the cells increase their weight, that is also termed as growth. If the cells the removing water increase their weight, that is also termed as growth. That is called dry weight. And it is amazing to know that one single root apical meristem of maize can give rise to more than 17,500 new cells per hour. See how crazy it is. A maize plant, the root apical meristem of the maize plant, RAM, root apical meristem, SAM, shoot apical meristem, not Samantha. Now it can give rise to, this RAM can give rise to more than 17,500 cells, new cells per hour. Are you understanding? Per hour. Okay, write it somewhere, it is important. And a watermelon can increase the size. Watermelon size, this much. Yeah, pochkai in Telugu. Sorry, I got my language. The watermelon, the big size watermelon can increase its size 350,000 times. That is called growth. But your pollen tube, growth of pollen tube is measured in the terms of length increase. Yes, isn't this correct? See, if this is your pollen tube, 1 minute, yeah, this is your pollen tube. Okay, let me write the stigma, style, ovary, etc. Typical flower, stigma, style and ovary. Inside there are some ovules, sepals, petals, etc. Yes, got it right. And now this is your typical flower diagram. This is a diagram of typical flower. Now what happens, watch. This is a stigma. A pollen grain is on the stigma. The pollen is right pollen. Now the stigma or the style allows it to germinate. Now it germinates. See pollen tube. Initially the pollen tube wasn't present, and after some time the pollen tube was germinating. After some time the pollen tube increased. After some time increased. Now here you can see that the length of the pollen tube is increasing. So growth of a pollen tube is measured in terms of its length. Okay, got it? Yes, correct. Yes, I'm going a little slow than yesterday's, so no problem, we can cover our session. Now let's see the phases of growth. As I said, we have 3 phases of growth. 1, meristematic. 2, elongation. I'll be telling you a trick how to remember this. Elongation, maturation. Before listening this trick, there is a small disclaimer. If you feel this trick, it is somewhat harmful or somewhat, I remember it like that. If you feel that it's somewhat wrong, please leave this trick. I am saying now only, please leave this trick. Meristematic phase. Compare meristematic phase into our young phase, our juvenile phase. While we are 10 years old, it is meristematic phase. Growing we are growing rapidly, but there is a phase of life where our rapid growth stops, that is some 16 to 18 years. During this phase our rapid growth stops, but we keep on elongating. Now we have completed our role on earth, we go to maturity. That is why I told you, if you want to get if you won't feel this trick is right, please read it. Marchuri or maturation. Or you can also say, we can compare the maturation stage. I have told you to remember, but compare the maturation stage of plant to our senescent stage, old stage, the 65 to 70 years average human, to that. Complete it. Young stage, meristematic stage 10 years, we go rapidly. Elongation stage 16 to 18 years in humans. This is just comparison. Please do not remember these values. This is just comparison. Why is this opening at all? I don't know, guys, what is happening to my tablet. Very poor tab. Yes, it is helping me in order to make videos. So that is it. Now let's move on. What is given in your running matter of your NCERT? The cells of this region. Which region? Where we are? Yeah, meristematic cell, elongation phase and maturation phase. Now searching for important points. Yeah, here the constantly dividing cells, both at the root apex and shoot apex, represent the meristematic phase of growth. Yeah, these apices which are present, the root apex and shoot apex, they have the meristematic cells, so that is why they show the meristematic phase of growth. What we have told about meristematic phase, it is a rapid divisioning phase. Why? Meristematic cells are present, that's it. Come on. The cells in this region possess a rich protoplasm, possess large conspicuous nuclei. You have to remember this. They can confuse you by asking inconspicuous nuclei. Their cell walls are primary in nature, thin cellulosic, with abundant of plasmodesmatal connections. Got it? This whole paragraph is very, very important, so that is why I am underlining it. Whole important. Please do not do like this in your NCERT. If your father sees, he will scold. Now the cells proximal, just next to this stage, are the present elongation or elongating cells. 1 minute. Next to proximal to the meristematic zone represent the phase of elongation. Increased vacuolation, vacuoles are developing. Vacuoles develop in this during this phase of life. Vacuoles develop during the phase of elongation. Cell enlargement, new cell wall decomposition are the characteristics of cells at this phase. Now further away from the apex, that is more proximal to the phase of elongation, there lies the portion of axis which is undergoing the phase of maturation. These have reached their old age. Now the cells in this region attain maximal size in terms of wall thickenings, protoplasmic modifications. Most of the tissues and cell types have been studied in chapter representative. So let's remove this, not important. Yeah, got it. Yeah, completed. Yeah, we have completed the phases of growth. So now let's move on to growth rates. Growth rates are very simple. We have two growth rates. Well, first of all, what is growth? It is um, increased growth per unit time. It is a growth per unit time. Remember, like it is a growth per unit time. Is termed as growth rate. We have two types. One is arithmetic growth rate. Second one is geometric growth. In arithmetic growth rate, see what is happening. In arithmetic growth rate, if one cell divides, here one cell, this is one cell, it is dividing like this, it forms two cells. In these two cells, one cell gets converted into the what we call what I'm saying, a permanent cell. One cell gets converted into permanent cell. So see this, this side of cells are permanent cells. No, this side, I hope. Yes. One cell gets converted into permanent cells, and these other cell, if this is a cell, guys, divided into two parts, it forms two cells. This is permanent. Now this keeps on dividing. This cell plus this cell, permanent cell, cells are being added. Now again this cell keeps dividing, this cell, this cell. So again here these two cells. So how many cells are being added? See, 1, 2, here 2 cells are being added to the plant body, and the remaining cells are dividing. Here 3 cells are being added to the plant body. But whereas coming to geometric growth rate, the cells do not differentiate, or you can say cells do not get matured, and these cells keep on dividing. One cell forms 2, 2 cells forms 4, 4 cells forms 8, 8 cells forms 16, 16 cells forms 32, 32, 64, 64, 64 plus 64, 128, 128, 512, 512 plus 512, how much? 512, 512, just for fun, guys, 1 minute, 4, 2, 10, yeah, 10, 24, 10, 24, 10, 24, 8, 4, 0, 2, 2, 0, 4, 8, 2, 0, 4, 8, 16, 1, 8, 9, 0, 4, again this, add them, simply completed. Okay, but whereas coming to zygote, this zygote shows both the phases. See, till here, from the zygote dividing till here, this phase is your geometric phase, and the rest of the life is your arithmetic phase. That is the embryo development, shows both phases. Completed. Just for fun I was doing like that, and we can represent this in a simplest equations. LT equal to L naught RT. LT is the length of time, length at a time t. L naught is the time at 0 or initial length. R is the growth rate or elongation per unit time. And if you try to plot this graph, it is gonna look linear. This is your linear graph. This is your linear graph. Okay, that is your linear graph, and this includes this part or portion is your rate. Now let's see that is the arithmetic growth rate. Sorry. This is the graph for arithmetic growth rate. Write it somewhere. Arithmetic growth rate, linear. Arithmetic growth rate is linear. So like this. Okay, like that. Now coming to your geometric growth rate. In geometric growth rate, we can see a S-shaped or sigmoid shaped curve, and we have three phases respectively: lag phase, exponential phase, and stationary phase. Lag phase, it is a slow phase, as the number of cells dividing are less, if the cells divide, form new cells, we have many number of cells to divide, so that is why the cells divide fast and increase their quantity, so that is called as exponential phase. Stationary phase, at a phase of life, these cells becomes constant and there is no more increase, so that is why the graph goes constant like this. Before it was constant, it suddenly increased, again it was constant. From constant to constant, sigmoid curve, this is your geometric growth rate and this is your geometric growth rate equation. W1 equal to W naught e to the power RT. E, it is called as base of natural logarithms. R, it is your growth rate, geometric growth rate, and it is also referred to as or efficiency index. It is also referred to as efficiency index. Why I am putting this many rounds, you can ask me, because it is very important and I have made a mistake in one of my exams. Efficiency index. Okay, got it, completed. Yes sir, yes Anna. Now we have something here. Decimate. Try to answer the question given in your NCERT. Read this paragraph, you will be getting the question and then try to answer. This is your HW paragraph. HW. Please mention in the comment box your answers. Now let's try to talk about the conditions for growth. See, during the plant cells elongation, we require some important conditions. The necessary conditions are water, oxygen and nutrients. This is again very important. I have made a mistake in one of my exams. Please remember these. Water, oxygen, nutrients. These three are only the very essential elements for growth. Yes, it includes light also, but light it comes in the later phases of life. Since the plants have to possess the ability or should get the ability of photosynthesis, then the light comes into major part. These are the three important and most precious essential nutrients for the plant growth. Temperature, light, they come in later phases of life, very essential elements for growth. Now they have explained you why these are essential here. Why oxygen is essential, helps in releasing metabolic energy. Why water is essential, it provides a medium for enzymatic activities. Why nutrients are essential, nutrients are micro and macro essential elements. They are required by plants for synthesis of protoplasm and acts as a source of energy. Got it? No. We have completed growth. Now let's discuss differentiation. Yes guys, you can feel that why I am too dull today. Yes, my health is not that good because I am having cold and I am not able to speak with full force. But nothing happens, let's continue. No problem, no one can stop us, no one can scratch. Let's discuss about differentiation. Differentiation, we have two types of differentiation: dedifferentiation and redifferentiation. The cells derived from root apical meristem and shoot apical meristems and vascular cambiums differentiate and mature to perform special functions. This act is leading to the maturation termed as differentiation. This is of two types: dedifferentiation, redifferentiation. What is redifferentiation? The living differentiated cells that by now have lost the capacity to divide, they can regain the capacity of division under certain conditions. In order to remember this, let me give you one trick to remember that. Redifferentiation. Okay, redifferentiation. 1 minute. Come here. come to this white space. Redifferentiation. In this redifferentiation, cutoff free, only differentiation happens. What happens in the redifferentiation is they would not get the ability to regain. There is no re in their life. Once they have differentiated, that's it's completed. No ability of regaining growth. Sorry guys, regaining growth once they have divided. But differentiation, they can possess the ability. What happens? How to remember this trick? Remember redifferentiation. In redifferentiation, what happens? Cutoff free. There is no differentiation. There is no ability of gaining the differentiation. Only differentiation happens. But as whereas a de-differentiation, they possess, they can again get back into their meristems. For example, intervesicular cambium. This is again very important. I have lost a mark here also. Why I am choosing this bloody heck pen? Intervesicular cambium and cork cambium. Very important. And redifferentiation, they lose the capability. Once they have lost the capability, they cannot divide. Let some tissues, etc., etc. Now we have told you, or I have told you, or NCERT have told us that growth in plants is open, that it is can be indeterminate and indeterminate. But can we say differentiation is also open? Yes, differentiation in plants is also open, because the cells or tissues arising out of the same meristem have different structures at maturity. What is differentiation? If this is the meristem, the cells arising out of this meristem, these 4 cells, if you divide at any plane, these cells possess the ability same to that of meristem, but they are just different structures. They are just different. They are diverging from one position. Okay, that is why the differentiation is also open in plants. Now we have completed growth and differentiation. Now let's see what is the relation between growth and differentiation. That is development. Growth plus differentiation gives you development. Growth plus differentiation gives you development. Sorry. And this is how growth happens in plants. It is just a diagrammatic representation. If you try to remember it. If you won't, please leave it. I have left that. I am not remembering. So what to do? No. Plants follow different pathways in response to the environment. Oh, let's try to underline with the black how it will be. Phases in their life forms and different kinds of structures. This ability is called as, oh wow, full of black. No need guys, no need. I thought you are able to see these. Yeah, plant follow different kinds of structures and it is called as plasticity. For example, heterophyly in cotton, coriander and larkspur. Here the juvenile leaves are a kind of like this. In coriander, this is an example, and the adult leaves are like this. They are showing plasticity. They can form different forms. They are showing different forms of life and different kinds of life in order to respond to the various stimulus. And we can see interestingly, and this development can also be seen in heterophilous development due to environment, can be also seen in response to the environment. This phenomenon is called as heterophilia, and it is an example of plasticity. And the example of heterophilia is your buttercup. See, in terrestrial habitat buttercup is like this, but in water habitat buttercup is like this. See how differences are there between these two. Oh, this is not coriander, this is larkspur, sorry, this is larkspur, this is buttercup. So it is due to environment. Here the environment is terrestrial, habitat is terrestrial, land. Here the environment is water. Here the environment is water. So if I color this, you are able to see it in green color. So let me show you my drawing skills. There you go with my drawing skills. Thank you for giving me first prize. Yes. Now let's move on. Why I am moving this fast? Yeah, that's your doubt. I hope completed this page. We are here. Okay. Development in plants, that is both growth and differentiation, is under control of intrinsic and extrinsic factors. The former means the intrinsic factors includes both intracellular or intercellular factors. Inter inside, intra outside, both. Okay. And these intercellular factors are your plant growth regulators, PGRs, or phytohormones. Now without any further ado, let's directly dive into the discovery. Before that, let's see the derivatives. So auxins are derivatives of indole 3 acetic acid, also called as IAA. Adenine derivatives, adenines are the derivatives of these what I am talking. Auxins are derivatives of IAA, auxinussu, and kinetins or cytokinins are the derivatives of adenine derivatives. Now carotenoids. Abscisic acid is a derivative of carotenoids. Gibberellic acid is the derivatives of terpenes. Ethylene, it is a gaseous PGR. And that's it. Now let's see the discovery of these PGRs using coleoptile. Interestingly, the discovery is accidental. All this is started by the observation of Charles Darwin and his son Francis Darwin. When they observed the coleoptiles of canary gas responded unilaterally illumination by growing towards the light source, phototropism. See, if this side was the light, if this is the side of light source, if here was the light, this is your sun, very bad sun. Yeah. Now what happened is these coleoptile tips bended towards the sun. Why it was happening like that? They got some doubt. There was something in this coleoptile tips. What they did is they cut the coleoptile tips. Then there was no bending. If they cut the coleoptiles of the canary grass, there was no bending. And if they cut this coleoptile and place it on an agar block, I hope this was done by 1 minute. I don't know, I just forgot who did this. Yeah, I hope Darwin and Darwin only. Yeah, Darwin and Darwin. No. Yeah, okay, leave it that Agarbach experiment. I was just confused who did it, so that is why I'm not teaching you. No. What does this Darwin and Darwin do? They put a small transparent or they have covered the scolioptile of canary grass with a cover. Then also they bend it towards light. This is because I'm telling you here only. This is because these auxins accumulate on this side. This is because of auxins. These are auxins. If this is the light source, this in this direction is the light source. Auxins, what they do is accumulate onto the surface opposite to the light source. So this side, they accumulate this side. Here they accumulate like this, auxins. So that is why there is a bending. There is a bending. And FW went for the first time. He has isolated auxins from the oat seedling coleoptiles. Now the Bacane or foolish seedling disease of rice seedlings was caused by a fungal pathogen called as Gibberella fusariae, and from this name only the gibberellic acid came from. E. Kurosawa reported the appearance of symptoms of the disease in uninfected rice seedlings when they were when they were treated with sterile filtrates of this fungus. This active substance were later identified as gibberellic acid. F. Spoog and his co-workers observed that from an internal segments of tobacco stems, this is important, the callus undifferentiated cells, and if this callus was put into the nutrient medium, it did not grow. To the medium containing auxins, it did not show growth. But if they put a nutrient medium like coconut milk, it showed growth. Coconut milk is not nothing, it is coconut water which we drink generally. It showed growth. So that stunned these Spoog and his co-workers. And what happened is Miller and Spoog then identified and crystallized this substance as promoting. It promotes cytokinesis, and they have named it as cytokinins. And the first thing to be discovered in the cytokinins was kinetin. Now in the mid 1960s, 3 independent theses. This is about ABA, abscisic acid. Now you have to remember these 3 names, it is very important. Abscisic acid in the mid 1960s has got the name inhibitor since it inhibits growth, inhibitor B, abscission 2, and dormin. These 3 were the names which were given by 3 independent scientists during the mid 1960s. Now Cousins confirmed the release of volatile substance from this was the discovery of ethylene. Okay, that is the discoveries. Now let's move on to the physiological effects of PGRs. I hope still more. How many pages are there? Still 6 pages are there. That 2 are of exercise pages. If you leave those 2, still 4 pages are there, guys. Okay, let's continue. Auxins. Auxin, from the Greek word auxin, to grow. A Greek word, auxins were derived from a Greek word called as auxins, which means to grow. The first isolated form urine. They were first isolated from human urine, and they were the derivatives of indole 3 acetic acid or IAA. So 1 minute, I am trying to underline without seeing. Yes. Indole 3 acetic acid. There can be 2 forms, one natural and synthetic forms. Natural auxins, let me write away. these examples or yeah, let's see here. Natural compounds having certain growth regulation properties. They are generally produced by growing epices. They are generally produced by growing epices. Wherever they migrate to the regions of action, auxins, here, IAA and IBA. IAA, IBA. These two were natural auxins. Synthetic auxins or NAA 2,4-D. Yesterday I have told you this 2,4-D while doing the MCQ series. We get this 2,4-D. It is a insecticidal application. Yes, guys. These were synthetic auxins. These auxins are used in extensively in agriculture and horticulture practices. What are the physiological effects? From here it is important. Let's change the style of marker. Yes, they initiate. What happened? Literally, I'm not understanding what has happened. Yeah, now it is changed. Okay, I have to click on done. What is this? They initiate rooting in stem cuttings, an application widely used in plant propagation. Auxins promote flowering, that is in pineapples. They promote fruit and leaf drop at early stages. Sorry, they prevent fruit and leaf drop at early stages, but promote abscission during the older mature leaves and fruits. Let me move my computer a little back. Yes, in higher plants growing apical bud or the lateral bud, sure. Yes, this is very important. Apical dominance, guys. What is apical dominance? Apical dominance, apical but dominance, dominating. If in plants, what happens is, if these auxins are present in plants, they allow apical dominance to happen. What is this apical dominance? Is if this apical bud is present and in the presence of auxins, they would not allow the lateral buds to grow, and they allow the plant only to elongate, but it cannot spread its branches far wide. In order to make these plants bushy, what they do is they cut the apical bud. If they cut the apical bud, there are no auxins there, so they can grow the lateral buds. They can give chance to lateral buds for growth. Okay, why 1 minute, guys. I wonder why is this black in color. Let me check the filters. I hope chroma key. Yeah, chroma key is present. Yes, now you get them perfect. Yeah, I was wondering why were these in black color. See, now we get them perfect. Yes, the chroma key was present, guys. The things which I teach lectures, it has chroma key. So these how what is happening here. Okay, remember that. And also, auxins induce parthenocarpy in tomatoes. Widely used herbicides 2,4-D to kill dicotyledonous weeds. Dicotyledonous weeds and does not affect mature monocotyledonous plants. Now let's revise the physiological effects of auxins which we have discussed. One, the thing which we have discussed is auxins promote flowering in pineapples. 2. Auxins promote apical dominance. Auxins induce parthenocarpy in tomatoes. They are used widely as herbicides for killing dicotyledonous weeds. 2,4-D is one of such example. And also we have seen that auxins, that's it, right? Yeah, root and stem cuttings, and they prevent fruit and leaf fall at early stages and promote abscission of mature leaves and fruits. Those are the physiological functions of auxins. Sorry. Yeah. Now let's move on to gibberellins. There are more than 100 gibberellins, and the GA3 was the first one to be discovered, and it is studied extensively. And these gibberellic acids uh, have wide range of physiological responses in plants. Their ability is to cross increase, this is very important, increase in the length of the axis is used to increase the length of grape stalks and sugar cane. Here they have forgotten the thing to write it here. Sugar cane only, sugar cane and grape stalks. I'm trying to see for buying an graphic tablet. I hope it is not fit for me. I cannot, you know, write without seeing on the thing which I am writing. So that is why I leave it apart. Sugarcane and the grape length is increased, and that is why it increases the what we call yield. Next one, I also they give a perfect shape in apples. They give a perfect shape in apples. Delay senescence, the delay senescence, and also malting process. Yes, today morning in MCQ session we have seen brewing industry malting process speed up. Next one, sugarcane. We have already completed this. So spraying juvenile conifers with GS hastens the maturity period, thus leading to the early seed production. Gibberellins also promote bolting, bolts. Remember this, bolts and nuts, bolting in cabbages, and many other plants with rosette habitats. So now once again let us read the physiological effects which have we completed. They try to increase the length of the plants, they increase the length of the grape stalks and sugar canes. Gibberellic acids delay senescence. They give a nice shape and elongation to apple. They promote malting process or speed up malting process in brewing industry. They promote bolting. Next one, spraying these GS on juvenile conifer hastens the maturity period and leading to the early seed production. Yes. Now these cytokinins. Now let's move on to cytokinins. Cytokinins have specific effects of cytokinesis and were discovered as a kinetin and modified form of adenine and purine. Adenine and purine derivatives are cytokinins. Cytokinin, adenine and purine derivatives. Now, from the autoclaving having sperm DNA. Kinetic does not occur naturally. Sorry, I'm getting sleep. I'm getting yawnings. Does not occur naturally in plants. Search for natural kinetic, and that led to giatin. Giatin occurs naturally in plants, and it was first discovered from corn kernels and same coconut milk. Naturally occurring cytokinins are synthesized in the regions where rapid cell division occurs. For example, root apex and shoot apex. It helps to produce new leaves, chloroplasts in leaves. So chloroplasts in leaves remain. Lateral shoot growth, adventitious shoot formation, etc. These cytokinins have competitive inhibition on apical dominance, and therefore they are competitive inhibitors of auxins. Sorry, I'm getting sleep. Yeah. let's move on. They promote nutrient metabolism which helps in delay of leaf senescence. Here what happens is, while the plant is reaching its senescence, the nutrients from the leaves you know uh, they mobilize to juvenile parts. So that is why they prevent this. If leaves nutrients are mobilized to juvenile parts, this leaf dies. So they prevent this, thereby delaying the leaf senescence. Hope you got it. Now let's move on to ethylene, but my wish is to teach you the photoperiodism, because we have already covered this abscisic acid ethylene in the regular classes, and we haven't covered this photoperiodism and the vernalization. So my thing or aim is to cover this first. Okay, ethylene and abscisic acid, please read that. I am getting sleep now only, and I am not able to take the lecture. I am not able to continue. So I will be ending the lecture by just talking about photoperiodism. Then we will be going. Now, what happens is, in some plants, the periodic exposure to light is necessary to induce flowering. If the plants are not exposed to light, there may not be flowering in some plants. In some plants, if they are exposed to light, there may not be flowering. So like that only, the plants which flower so fast that without the help of light, without the exposure of more light, they are termed as short day plants. So the plants which require a long exposure to light, they are termed as long day plants. The plants which can flower both when they are exposure to the photo light either shortly or to a little extent or to a more extent, they can flower. So for talking this, we require a word called as critical photoperiod. This photoperiod line, it is the differentiating line in some plants in order to induce flowering, is called as critical photoperiod. If the plants, if in the long plants, if these plants were exposed more and more to this critical photoperiod, if they are exposed more and more to this critical photoperiod, then these plants flower. Here you can see the plants are flowering above the critical period. Below the critical period, there are no flowering. But in these plants, if you talk about these plants, below the critical photoperiod, there is below the critical photoperiod, there is no flowering. But the above the there is flowering in short day plants, but above the critical photoperiod, there is no flowering. So these are called as short day plants. Remember one thing: more exposure to light, no flowering, short day plants; more exposure to light, flowering, long day plants. You remember only either way. Either you remember when there is no flowering, or remember when there is flowering. Please do not try to focus on both, then we lose everything. Okay, try to only remember and concentrate only one thing. When there is no flowering, okay, well and good. If they are more exposed to light, there is no flowering in short day plants. If they are less exposed to light, there is no flowering in long day plants. Or try to remember flowering. If they are exposed to more light only, there is flowering in long plants or long day plants. If they are exposed to short day or short period for light or below the critical photo period only, they flower. Those are called as short day plants, and flowering is seen any time after vegetative maturity in the day neutral plants. Okay, in the day neutral plants. I am searching for the examples. There is no example here. open the speech. Okay, not only here, see this is very important. Not only the duration of light period, but the duration of dark period is also equally important, and that hence it is said that flowering in certain plants depends not only on the combination of light and dark exposures but also on the relative durations. This response of plants to the day and night is termed as photoperiodism. That is the definition. That is the definition. Got it? Got it? Yes, Anna. Yes, sir. And later it was discovered that this is due to, it was hypothesized that this is due to an hormonal substance that is migrating from leaf to shoot apices when the plant needs to flower. So that what was discovered and hypothesized later. Now, I hope we have completed photoperiodism in our regular classes or in our detailed videos. We haven't completed vernalization. Now only I got it. When I was saying about the diagram and critical photoperiod, then I got it. Oh, I have covered this one. Why am I again telling you what to do? And that's it. If we talk about vernalization, we complete. There are plants for which flowering is either qualitative or quantitatively dependent on exposure to low light. This phenomenon is termed as vernalization. What happens is, in some plants the flowering is directly dependent upon low temperature exposure. If the plants are present at during low temperatures, then they flower, that is called as vernalization. It prevents precious reproductive development late in the growing season and enables the plant to have sufficient time to reach maturity. Now what happens is, it prevents the precocious reproductive development late in the growing season. In vernalization, what happens is, the plants do not achieve reproductive development in the growing season, but it enables reproductive maturity only once after the growing season is completed and plants they're ready to flower. Growing season is completed and then only they are able to mature reproductively. That is called as vernalization, and it refers to specially to promote flowering by a period of low temperature. Some important food plants like wheat, barley, these are very important. Some important food plants like wheat, barley, rye have rye, that is a plant name. I don't know what's that plant. Different kinds of varieties in winters and spring varieties. We have two varieties, as vernalization plays an important role in them. The low temperature plants are there, and medium temperature plants are there, and high temperature plants are there. The winter plants have the low temperature. The spring varieties normally planted in the spring came to flower and produce and gain before the ending of the season. However, in winter varieties, however, planted in spring, it normally fail to flower and produce mature grains within the span of a flowering season. Hence, they are planted in autumn. They germinate and come to small seedlings and resume the growth in spring. Now, what happens is, as this vernalization shows an effect on low temperature, we have two types of varieties of some important food plants like wheat, barley, and rye. These plants have spring varieties and winter varieties. In spring varieties, what happens is, plants come to flower, they gain reproductive maturity and they come to flower before the growing season, as temperatures are high. Vernalization is not effective that time. But whereas coming into winter season, winter varieties, these varieties in winter there is sorry for that sound. In winter there is low temperature and vernalization shows an effect on winter varieties. So in order to as our vernalization shows effect on winter varieties, what happens is these winter varieties once planted, they would not come to flower in winter. As vernalization plays a key role, it does not allows the plant to develop reproductively in the growing season. Only after the growing season only vernalization helps the plant to grow reproductively. That is why they cannot flower, and hence they cannot produce germinate or fertile seeds. That is why they are planted in autumn. In autumn, a season before the winter, and as in winter vernalization plays an important role. They can produce small seedlings and resume growth in spring and harvested around in midsummer. There vernalization does not play, because in spring, when converting from winter to summer, there is a mid period called as spring. Here the temperatures rise a little and vernalization decreases, and plants can flower and plants can produce seeds, and they can be harvested in summers, mid-summers. So that is why it is an example of vernalization. Hope you are understanding it. If you haven't understand, let me tell you once again. These are some important food plants like wheat, barley and rye show have effects of vernalization. So that is why there are two types of varieties: one spring variety, two winter varieties. Spring, it is a time period from, or it is a weather, or it is a season from winter to summer. It is a change of phase from winter to summer. That is why in spring temperatures rise a little when compared to the summer, and vernalization plays an important role at low temperatures. Now in spring varieties there is a high temperature, and the vernalization would not play an important role, and that is why they grow, they reproductively mature, and they produce flowers, they give seeds. But whereas coming into winter varieties, once they are planted in winter, this vernalization would not allow them to flower. It gives a sufficient season for growing. It would not allow them to flower in winter, and throughout the season there are no flowers. What happens then? Throughout the season there are no flowers. So that is why what happens is, when these winter varieties are planted in spring, when they are planted in winter, they grow in spring, they grow in what I am telling you, if they are planted in winter, they come to flower in spring, and they can be harvested in mid summers. Got it? And another example of vernalization is seen in biennial plants. Biennials are monocarpic plants that normally flower or die in the second season. 1 minute, I'll be just back. Now another important example for vernalization is your biennials. What are these biennials? Biennials are the plants. Now what are these biennials? Biennials are the plants which flower in one season, and as soon as they flower in this season, in the next season they die. In them also vernalization plays an important role, and common examples are sugar beet, cabbages and carrots are some common examples of biennial plants. So in these plants such as these biennials, sugar beet, cabbages and carrots, also the vernalization plays an important role. That's it guys, we have completed this, and I have some important point here in summary. So the important point is, growth is one of the most conspicuous events in living organisms. So remember that, they can ask you the statement in your exams. So that's it guys, we have completed successfully the NCERT series of the plant growth and development chapter. You have to only read about the ethylene and ABA, abscisic acid. Tomorrow morning you will be having the MCQ series, that is 30 under 30 MCQs. Then meet you tomorrow there in that session. So thank you guys, this is Vamsi signing off from Biolab, and this have a great day further. And if you like the video, please hit that like button and also subscribe to our channel Biolab. And this is Vamsi signing off. Have a great day. Thank you guys.